Greetings, I'm Dharshini, Parmal, graduate student under Dr. J. Azrain J. Abdullah from Department of Physics, Faculty of Science, University, Putra, Malaysia. On this glorious day, I would like to present on my research titled Green Synthesis of Reduced Graphene Oxide and Cytotoxicity Studies. Green nanotechnology has gained attention among the researchers from various fields to work on environmental sustainability materials. Herein we employ oil palm leaves in producing reduced graphene oxide, a graphene-based nanomaterials for biomedical application. Reduced graphene oxide is considered to be an attractive route to obtain graphene-like behavior and holds useful properties. We have selected the biomass waste of oil palm agro-industry, which is the largest contributor of biomass in Malaysia. Oil palm biomass are constantly generated in large quantities annually, with a small fraction being converted into value-added products while a large percentage are left underutilized. Thus, our current research extends the effort and focus towards the usage of local biomass waste such as plant-based extract as a reductant. Moving on to the research methodology. The research methodology of our study starts with the preparation of graphene oxide using improved Hermes method via oxidation and exfoliation of graphite powder. The as-prepared graphene oxide was subjected to grain reduction with the use of oil palm leaf extract under reflux. The synthesized reduced graphene oxide were characterized for further usage. The toxicity studies were performed using brine shrimp assay, whereby graphene oxide and as-prepared reduced graphene oxide under different concentration were examined. Each exposure was carried out by triplicate and the density was 10 cis per well in each replicate. The plates were incubated at room temperature under a continuous illumination. The hatchability of the cyst in each concentration was monitored under a microscope. The results of main characterization performed were as listed. FTIR were conducted to identify the biomolecules that are responsible for capping, reduction, and stabilizer. Based on the peaks listed in Table 1 shows that graphene oxide exhibits peaks at the wave number 3220, which is assigned for the hydroxyl group. Other peaks centered around the wave number 1731, 1621, 1364, and 1220 and 1043 are assigned to carbonyl, carboxyl, epoxy, and aromatic groups respectively. It is evident that in reduced graphene oxide, the intensity of oxygen-containing functional groups are nearly invisible, which is suggesting effective reduction of graphene oxide. Atomic structure of the sample were determined by using XRD. Figure 1 shows the XRD pattern of graphite, graphene oxide, and reduced graphene oxide. Figure 2 shows the Raman spectra of graphite, graphene oxide, and reduced graphene oxide. Image 1 shows the TEM image of graphene oxide and reduced graphene oxide. And figure 3 shows the graph of hatching percentage versus concentration of graphene oxide and reduced graphene oxide using brine shrimp assay at different time point, which is 18 hours, 24 hours, and 48 hours. Image 2 shows the Atima Selena image, which is the brine shrimp image in graphene oxide and reduced graphene oxide solution. Discussion. The XRD pattern confirms the graphene oxide and reduced graphene oxide was successfully formed with the presence of peaks at 2 theta for graphene oxide at 10.5 degrees and reduced graphene oxide at 24.5 degrees. Raman spectroscopy results reveal two prominent peaks, which are D and G band. The D band of reduced graphene oxide shifted to 1353 from 1355 wave number in graphene oxide. Meanwhile, G band of reduced graphene oxide shifted to wave number 1603 from 1605 in graphene oxide. The ID and IG ratio of reduced graphene oxide and graphene oxide did not show any significant differences. The TEM image shows the folds and curls on the surface of graphene oxide, while the reduced graphene oxide surface was well restored and improved. Brine shrimp-based toxicity is a quick and easy test for early indication of nanomaterial toxicity. Toxicity studies reveals that reduced graphene oxide is less toxic compared to graphene oxide. This reduced graphene oxide shows excellent biocompatibility on brine shrimp. In conclusion, a simple yet cost-effective and stable plant-derived reduced graphene oxide was successfully prepared. Our future work will be on the incorporation of silver nanoparticles and reduced graphene oxide for potential synergetic effect, which may open a new avenue for cancer treatments. These are references for my poster presentation. Thank you.